Once again, welcome back with another video lecture. In this video lecture, the topic under discussion is bacteria. Bacteria are most abundant microorganism on earth. They are prokaryotic, microscopic, unicellular microorganism that lack membrane-bounded cell organelles. The bacteria are classified based on cellular morphology. The general appearance of individual bacterial cells under bright field of compound microscope is known as cellular morphology. So bacteria mainly in three forms, that is cocci. These are round or circular bacteria. Coccus is for single cells. Then comes bacilli. These are rod shaped bacteria or cylindrical shape. Bacillus for single cells. Then spirilla or twisted rods or winding bacteria. Spirillum for single cells. A typical bacterial cells measures 2 to 8 micrometer in length, and they forms associations such as pairs, chains, tetrads, sarcina or clusters. Now here are shapes that a bacteria can take up. Diplococcus, round bacteria that remain in pairs after reproduction. For example, Neisseria, gonorrhea, they have single plane of division. Then a bacteria can take up shape. Streptococcus, when round bacteria remain in chain form. For example, Streptococcus pyogenes. Next, a bacteria can take up shape of tetrads, which is clusters of 4 round bacteria. For example, Micrococcus species. Tetrads have double plane of division. Also, a bacteria can take up shape of sarcina, in which bacteria are found in cuboidal arrangements of 8 cocci. For example, Sarcina ventriculi. Next, a bacteria can take up shape of Staphylococcus. It forms irregular grapes-like clusters which have random plane of division. Then second category, what's called bacillus or rod shape. When bacilli appear in pairs, we called it diplobacilli. Just like Morphology bovis bacteria, when bacilli appear in chain form, known as streptobacilli. For example, Streptobacillus notomitis. Then comes Coccobacillus. When bacilli bacteria resemble cocci, we called it Coccobacillus. They are short, rod or oval. Examples is Haemophilus influenza. Then comes Spirillum. Spirillum may have in 3 forms, that is spiral or helical with thick cell wall. Example is Spirillum volutum. Next is spirochete. They are spiral shape with thin cell wall, just like Treponema pallidum. A bacteria may also can take up shape of comma or vibrio, just like Vibrio cholera. Some bacteria have what's known as flagella, which are thin rigid filaments, and these allowed for bacterial motility, self-propulsion and movement. They are made up of proteins, and these proteins can move and propel bacterial cells forward. Some bacteria have flagellum at one pole called monotrichous, just like Campylobacter species. A bacteria which have bunch of flagella at one end known as Lophotrichous. For example, Helicobacter pylori. Also a bacteria having single flagellum on both ends called Amphitrichous, just like Alcaligenes facialis. When bacteria have flagella in all surface, known as Peritrichous. For example, E. coli. Next comes Pili. Singular is pilus. Pili are A glycoprotein appendages or finger-like projections lies on bacterial cells and are shorter and thinner than flagella. The Pili have special role in bacterial conjugation. Bacterial conjugation is the transfer of genetic material from one bacteria to another. By forming a bridge using Pili, this exchange of genetic material can be in between bacteria and host cell or from one bacteria to another bacteria. Then a bacteria have Fimbriae. Bacteria have three types of appendages, that is flagella, Pili and Fimbriae. Fimbriae are glycoprotein appendages on bacterial cell. They are shorter than Pili and used for attachment to contact surfaces. Fimbriae have ability to cause a disease by bending to a surface of cell in our body. Singular is Fimbria. Then comes capsule. Many pathogenic bacteria produce an extracellular enzyme to synthesize a polymer that form a layer around bacterial cell. This layer is called capsule. Capsule is a virulence factor for bacteria and made up of polysaccharide which protect bacterial cell from phagocytosis, where phagocytosis is the ingestion of bacteria by phagocytes, while phagocytes are the cell that protect the body by ingesting harmful foreign particles. The capsule is found in most commonly gram-negative bacteria, for example Escherichia coli in some strains, and also Neisseria, menengitidis and Klebsiella pneumoniae. Then comes cell wall, which is made up of peptidoglycan. The cell wall are in between plasma membrane and capsule. The peptidoglycan is a mesh-like structures surrounding the entire cell, which is made up of polysaccharide chains cross-linked by peptide, that is NAM and NAG, N-acetyl muramic acid and N-acetyl glutamic acid. The cell wall enables the bacteria to classify into two groups, that is gram-positive bacteria and gram-negative bacteria, because when we dye them they appear in different colors which totally depend on cell wall composition. So what are the different composition of cell wall? The gram-positive bacteria have thick cell wall because of multi-layers of peptidoglycan, while gram-negative bacteria have thin cell wall because they have less layers of peptidoglycan in their cell wall as shown in the diagram. Also, in addition to this, gram-negative bacteria have another outer membrane which are notable between gram-negative and gram-positive bacteria. So because of this difference they appear in different colors after gram staining. Then comes plasma membrane. The bacteria innermost layer is known as plasma membrane. Plasma membrane also found in human cells. The plasma membrane is semi-permeable membrane which allowing transportation of substance in and out of the cell. In bacteria, the plasma membrane or cell membrane is composed of phospholipids, which is double layer or bilayer. It contains proteins embedded or integrated in membrane. This plasma membrane have hydrophilic region, which mean a region that loves water, and also a hydrophobic region. Hydrophobic region mean water scare, and this character of phospholipid basically enables it from the phospholipid double layer or bilayer. Semi-permeable, which mean it restricts the kind and amount of molecules that enter and leave the cell. Then comes ribosome. So ribosomes are the site of protein synthesis, or it is a factory of protein synthesis. Ribosomes are bodies of RNA and proteins. They are associated with protein. Here we have prokaryotic ribosomes and eukaryotic ribosomes. Prokaryotes have 70 S ribosomes, while eukaryotes have 80 S ribosomes. They have large subunits and small subunits. They are made from proteins plus RNA fragments. 50S or large subunit have 5S RNA, 23S ribosomal RNA, and 31 protein, while 30S have 16S RNA plus 21 proteins. Then comes plasmid. Bacteria also have an additional genetic material called plasmid. They lies in the cytoplasm and are not the part of circular DNA. They are an extra chromosomal rings of DNA. They have additional genetic information and can be incorporated in circular DNA, the large plasmid, and they can be transferred between two bacteria, the small plasmid, through bacterial conjugation. They have antibiotic resistant genes and are very important in genetic engineering. Then comes mesosomes. Mesosomes is an invagination of cell membrane which helps in cell division through binary fission. Next is nucleoid region. The DNA of bacteria is circular, not linear. Such is the DNA in humans. But it must be noted that humans also have circular DNA in the mitochondria. Anyhow, the bacterial DNA is circular and the nucleoid is a region in cytoplasm where bacterial chromosomes and DNA are located. Bacterial nucleoid are not membrane bounded nucleus but simply an area of cytoplasm where the strands of DNA or chromosomes are found. Thanks for watching. Don't forget to subscribe, like, share and comment.